IB Bio, Human Physiology Part 7 is the first of two movies that cover defense against disease. Human Physiology Part 7, Defense Against Disease A. This movie will focus specifically on the nature of pathogens, disease-causing organisms. How pathogens gain entry to the body will be covered, as well as some non-specific adaptations of mammals to keep pathogens out of the body. The essential idea is the human body has structure and processes that resist the continuous threat of invasion by pathogens. This slide and the next provide the outline for all of the movies available for Topic 6 Human Physiology for SL and HL students. Use the outline to find the movie you need for review. This movie is focused here. Here is the second slide of the outline for all movies of Topic 6, Human Physiology. A pathogen is a disease-producing organism. You can see in this image that a bacterium, a possible pathogen, is being ingested by a phagocyte in green. White blood cells, known as phagocytes, will identify invading pathogens and ingest them. Pathogens, as disease-producing organisms, are simply pursuing the nutrition that they need to sustain life. When pathogens infect us, or this gorilla, they do us harm while doing well for themselves. This is the circle of life. The gorilla is not a pathogen, but pathogens of mammals include organisms from all the kingdoms except plants. Disease and its pattern of infection can be studied through a branch of science known as epidemiology. We could study the epidemiology of a pathogen making its way through a gorilla population. Pathogens gain entry to the body through an open wound, through the respiratory tract, through the digestive tract, through the reproductive tract, through insect or animal bites, or through the placenta. Pathogens gain entry through the water we drink, the air we breathe, the food we eat, and through open wounds where the protective skin has been breached. As well, pathogens gain entry when insects or other animals bite us. Pathogens gain entry to a fetus through the placenta from mother to fetus and through sexual contact. HIV is a sexually transmitted disease. Pathogens of mammals originate from all biological groups except plants, viruses, the prokarya domain, fungus, Protista, and Animalia. Viruses are small biological particles, technically not living, as they require a host cell to reproduce. Viruses are unable to reproduce without a host cell. As a result of the fact that they are not composed of cells and do not have the metabolism of a cell, antibiotics are ineffective against viruses. Examples of pathogenic viruses include HIV, smallpox, influenza, rabies, dengue fever. Here's our first relevant IB syllabus statement. Explain why antibiotics are effective against bacteria, but not against viruses or eukaryotic cells. Remember, bacteria are prokaryotic cells. Now, historically, some bacterial diseases were fatal. Pneumonia, gangrene, syphilis. But with modern antibiotics, some bacterial disease have been nearly eliminated, such as smallpox or polio. Now, viruses lack a metabolism and cannot therefore be treated with antibiotics. And antibiotics block processes that occur in prokaryotic cells, but not eukaryotic cells. Viruses, one of which is seen here, are not surrounded by a cell membrane, and they use metabolic pathways of the host cell to reproduce. Thus, antibiotics, which block cellular metabolic pathways, do not function with viruses. Viruses lack a metabolism and cannot therefore be treated with antibiotics. The smallpox virus was the first infectious disease of humans to have been eradicated by vaccination. Here is an image of influenza virus particles. Viruses lack a metabolism and cannot be treated with antibiotics. Antibiotics block processes that occur in prokaryotic cells but not in eukaryotic cells. Influenza is a viral pathogen. Specific proteins on the surface allow the virus to enter the host cell, and once replicated, allows the virus to exit the host cell. Viruses lack a metabolism. They use host cell metabolism, and thus viruses cannot be treated with antibiotics. HIV is a viral pathogen. HIV is a retrovirus with an RNA core rather than DNA. The RNA is transcribed within the host cell into mRNA for viral protein production. RNA viruses, like HIV, also have the enzyme reverse transcriptase, so that viral RNA can be copied into DNA. A virus lands on the surface of a host cell. It inserts its DNA or RNA into the host cell. The host cell, in this case a bacterium that's been infected, incorporates the viral DNA into its own DNA and then proceeds to make new virus. The new virus particles increase in number and then burst out of the host cell, killing the host cell. And this process is called infection. Retroviruses, RNA viruses like HIV, gain entry into a cell. Then the RNA is replicated to make new viral RNA, or it's transcribed to make viral protein. Then the protein with the newly replicated RNA join to make new viral particles that exit the cell. What we see in this image would generally be called viral infection. Here's another look. A virus lands on the surface of a host cell, a bacterium in this case, and inserts its DNA into the host cell. The host cell incorporates the viral DNA into its own DNA, then proceeds to make new virus. The new virus particles increase in number and then burst out of the cell, killing the host cell. So while pathogens can be viral, pathogens can be prokaryotic, bacterial, such as tuberculosis, staphylococcus, streptococcus, clostridium, and chlamydia. And antibiotics are effective against prokaryotic pathogens. Here are rod-shaped bacteria piled up on a surface. Here are bacteria that cause tuberculosis being engulfed by a phagocytic white blood cell. Antibiotics target cell metabolic processes in these bacteria, such as the formation of cell wall. In this X-ray of a human lung, we can see the tubercles of the Mycobacterium tuberculosis forming in the lung. As bacteria are living cells with independent metabolic processes, antibiotics work to kill pathogenic bacteria. Penicillin and other antibiotics have been very, very important to humanity. Unfortunately, some strains of bacteria have evolved with genes that confer resistance to antibiotics, and some strains of bacteria have multiple resistance. Howard Florey, Ernst Boris Chain, and Alexander Fleming shared the 1945 Nobel Prize in Physiology or Medicine for their role in the development of penicillin. Although Fleming received most of the credit for the discovery of penicillin, it was Florey who carried out the first ever clinical trials in 1941 of penicillin at the Radcliffe Infirmary in Oxford on the first patient, a postmaster who lived near Oxford. The patient started to recover, but subsequently died because Florey had not made enough penicillin. Florey and Chain's tests on the safety of penicillin would not be compliant with the current protocol on testing. Fleming began his work on antibacterial substances after the First World War, during which he had witnessed countless deaths, countless soldiers' deaths due to bacterial infection of wounds. In 1928, Fleming was in the midst of investigating a genus of bacteria called Staphylococcus, and it was during September of that year, 1928, that he made a serendipitous discovery. The story goes that Fleming was somewhat messy in the lab and had returned from a vacation to find many of his bacteria culture dishes had been contaminated with mold. He later realized that on one of the dishes, the mold colony had a clear zone around it where the bacteria could not grow. Fleming identified the mold as penicillium, and he isolated an extract from the mold and named it penicillin. He spent some time investigating its antibacterial effects, published his findings to surprisingly little fanfare. He subsequently found that cultivating the mold and extracting penicillin was a laborious task, and because of this, and his conviction that the agent could not work effectively in the human body, he eventually abandoned his work. Then, in May of 1940, Florey inoculated 8 mice with Streptococcus bacteria. 4 were given doses of penicillin. Within a day, the mice without the penicillin had died, while those given penicillin lived. It was Florey who carried out the first ever clinical trials in 1941 of penicillin on the first patient. The patient started to recover but subsequently died because Florey had not made enough penicillin. Remember, Florey and Chain's tests on the safety of penicillin would not be compliant with current protocol on testing. Ernst Chain was the driving force behind isolating and testing penicillin, but when it came to manufacturing penicillin on a large scale, it was Howard Florey who made more of a contribution by locating several U.S. companies who were willing to provide resources when UK factories could not because of the Second World War. Mass production of penicillin began in the U.S., and by the end of the war, U.S. companies were making 650 billion units of penicillin every month. Moving away from prokaryotic pathogens, we come to protistan pathogens. The protistan kingdom has species that are pathogenic toward people. Malaria is a nucleated unicellular pathogen, the cells of which are seen here. Malarial cells are transmitted to people by the mosquito. Trypanosomes are pathogenic. Both malaria and trypanosome protists are heterotrophs. The fungus kingdom also has species that are pathogenic toward people. Athlete's foot and ringworm are 2 examples. This bat in the lower right corner has a pathogenic white fungus on its nose. The animal kingdom produces pathogens as well. Tapeworm, guinea worm, hookworm, and ticks are all pathogens. So that brings us to the next IB syllabus statement. Outline the role of the skin and mucous membranes in the defense against pathogens. State that skin and mucous membranes form a primary defense against pathogens that cause infectious disease. The skin and mucous membranes would be part of our non-specific defense systems that serve to deter any pathogen or really maybe no specific pathogen. We'll look at specific defense mechanisms in the next movie. The skin and the mucous membranes are included with external non-specific defense mechanisms. The clotting of blood to close wounds is also an external non-specific defense mechanism. We also have non-specific internal defense mechanisms. These include white blood cells that are called phagocytes, the inflammatory response, and proteins that bind pathogens called complement proteins. The skin on the left and ciliated epithelia on the right. Ciliated epithelia, such as along the trachea or the oviduct, provide an external barrier to the entry of pathogens. Stomach acids kill pathogens that have entered by food and water. Glands along the skin also produce oils that inhibit pathogens. In this image, we can see a breach in the skin with the introduction of bacteria. Histamines are released by mast cells that reside in connective tissue all over the body. The histamines cause blood vessels to widen, bringing phagocytes to the damaged area. Phagocytes engulf bacteria through phagocytosis. You can see that happening here, and they digest that bacteria inside the cell with lysosomes. As well, a break in the skin that causes capillaries to open, bleeding, in other words, that would require a clot to close the wound. Here's the relevant IB syllabus statement related to blood clotting. State that cuts in the skin are sealed by blood clotting. explain the process of blood clotting, including platelets, clotting factors, thrombin, fibrinogen, and fibrin. Platelets that rupture at the site of injury release clotting factors known as thrombokinase. It's an enzyme, and it catalyzes the formation of thrombin from prothrombin. Prothrombin is a protein that circulates in the bloodstream. Thrombin catalyzes the formation of fibrin, an insoluble protein, from fibrinogen, a soluble protein in the blood. In this image, we can see the fibrin fibers with red blood cells. This is the beginning of a clot that closes a wound that is a possible entry point for pathogens. Red blood cells, fibrin, seen in green in this image, and platelets will form the plug as the basis of a clot that seals the wound, reducing the likelihood of pathogenic invasion. Remember from our study of the circulatory system that blood clots can form in coronary arteries, as you can see here, causing occlusion of the coronary artery. This is called coronary thrombosis, and it's the start of what might be a very dangerous heart attack. The coronary arteries supply heart muscle with oxygen and glucose. Coronary thrombosis is the formation of a blood clot along with plaque in the coronary artery of the heart. This blood clot restricts blood flow so that all the heart muscle cells below the clot are deprived of blood. This would result in a heart attack. So, just to recap, as a part of non-specific defense, I've already mentioned the skin, the ciliated mucous membranes, and clotting. As well, I've mentioned the inflammation that occurs due to histamines and the dilation of blood vessels that bring phagocytic white blood cells to the site of an injury. Now, don't forget that stomach acids also serve a role in deterring pathogens from gaining entry through food and water. So here's an image that I put together for non-specific defense. The skin, ciliated mucous membranes, and stomach acids are all non-specific external defense mechanisms. Once a pathogen enters the body through a wound, phagocytes arrive in the circulatory system that will engulf the pathogen and digest it. The blood clotting process, prothrombin to thrombin, fibrinogen to fibrin, will seal the wound. As a result of the injury and the entry of bacteria, the mast cells release histamine, and the histamine causes blood vessels to dilate, typical of an inflammatory response. The dilated blood vessel brings numerous phagocytes to engulf the pathogens through phagocytosis, digesting the pathogen with a lysosome. So here's a relevant IB syllabus statement. Outline how phagocytic leukocytes, white blood cells, ingest pathogens in the blood and in body tissues as part of non-specific defense. In this image, it appears the wound is healed and the remaining pathogens are engulfed by phagocytes through phagocytosis. Here's an image to better understand phagocytosis. The pathogen is engulfed, and then the vacuole is joined with a lysosome. The enzymes within the lysosome digest the pathogen, and the undigestible wastes of the pathogen are released through exocytosis. Here's a micrograph of a phagocyte engulfing a number of pathogenic bacteria. Here is another micrograph of a phagocytic white blood cell engulfing a pathogenic bacterium through phagocytosis. In this micrograph, we can see phagocytic white blood cells, sometimes called macrophages, engulfing spherical bacteria. I would like you to bring the term macrophage into your vocabulary. It's a large white blood cell that ingests viruses or bacteria by phagocytosis. In this micrograph, we can see a phagocytic white blood cell, sometimes called a macrophage, engulfing Here is another micrograph of a macrophage approaching a pathogen. Macrophages are resident in the alveoli, the liver, the kidney, and the brain, just to name a few locations. Inflammation, something you have all experienced when infected, involves the following set of events. First, there's the injury. We have damaged cells. Mast cells release histamine. Histamine causes blood vessel dilation, increased capillary permeability, and phagocytes arrive. And kinins are released from injured cells so that nerve endings are sensitized and you feel a bit of pain from the infection. As we close in on the end of the movie, I want to end with a look at blood cells. Blood cells form in the bone marrow as stem cells ready to differentiate into any one of various types of blood cells. Red blood cells, platelets that are important in the clotting process, and many types of white blood cells. All of the white blood cells on the right side of this diagram are phagocytes that ingest pathogens. The monocytes become the macrophages that I've spoken of. And the lymphocytes are white blood cells that I've not yet mentioned. Lymphocytes play a role in the specific defense system, which is the subject of the next movie. You've probably heard of B cells and T cells. Stay tuned. Here's a slide that provides some perspective on blood cells. Red blood cells are found in the 5 millions per cubic millimeter, while white blood cells are found in the 5,000s per cubic millimeter. Macrophages and lymphocytes, the B and the T cells, play a role in the specific defense systems. You should know the role of platelets in blood clotting. So this slide has been an organizing outline for this movie. Hopefully everything here makes sense except for complement proteins, about which you do not need to know for IB Bio. The subject of Defense Against Disease B, the next movie, will be here with the B cells that make antibodies and the T cells that activate B cells and search the body for cells infected with viruses. And that brings us to the end of Topic 6, Human Physiology, Part 7, Defense Against Disease A.